So I wasn't quite sure what level you were at. So if I'm talking at too basic a level on quantitative genetics, let me know and we can move along. Otherwise, I actually can make these PowerPoints available to anybody who's interested in them if we can figure out how to post them. So basically what I thought I would do is start by just giving an overview of how quantitative genetics works. And perhaps this is what might be too basic for some people. Oops, I didn't mean to have that in there. First of all, we should talk about what evolution is. And this is something that people make mistakes about all the time. This is an old definition that I got out of a biology book, which is all the changes that have transformed life on Earth from its earliest beginnings to the diversity that characterizes it today. This is really fundamentally incorrect because it's defining evolution in terms of its consequences, not in terms of the process of evolution. There's a bunch of different definitions that we'll run into. Lasting change in mean phenotype of a population that transcends the life of an individual. That's one of my favorite ones. Change in gene frequency. Most of the time that's a good definition, but there's times where it falls apart. And the heritable change in the mean phenotype of a population. All of those are definitions. Now what you'll notice that are valid, what you'll notice about these is it's about change taking place in the moment. It's about how the phenotype changes, how a population changes over short periods of time, and it's not anything about the consequences. And so this is actually my thought on the definition. Evolution is a change in the population due to birth and immigration and death and emigration. So again, a very straightforward, change in the distribution of a population, specifically excluding things like development. And so, for example, if we look at, this is the European corn borer, and when they overwinter as larvae, eventually they become pupae and moth, and then the adults, and then the adults lay eggs, and they go through several cycles. So if you sample the population at any different time, you may discover that everybody is a worm to be. Or everybody is an egg, or everybody is a moth. And so clearly the population is changing over time. But it's not changed due to the birth and death of individuals. It's changed due to development. So we don't want to call that evolution. Okay, so this is my definition. Change in the distribution of phenotype of population due to the gain and loss of individuals. So, where does quantitative genetics fit into this? Basically, quantitative genetics is about change in phenotypes. This means that there must be heritable variation in the populations, and quantitative genetics is the study of this variation, is the study of variation in populations and the evolutionary consequences of that variation. And it's going to focus on heritable variation, which in almost all cases is the underlying genetic effects. And I always put that caveat in there because there's always somebody who's interested in, for example, humans, and they have culture. And it gets very awkward when we move to something like humans because, in fact, you speak the same language as your parents did. In a very real sense, that acts as a heritable variation. And clearly it's not genetic. And so in theory, this cultural transmission could fit in here as well. So in a very real sense, quantitative genetics is the study of evolution. Okay. Okay, just to remind you, Mendelian genetics exists. And what we want to do is we understand how genes work. And so what we want to do is we want to build a foundation based on our understanding of Mendelian genetics. And here I just put up a standard cross that you're familiar with, where you get the 3-to-1, standard 3-to-1 variation and stuff like that. And the point is we want to build from this foundation into the variation we see, because Mendel was extremely lucky. He has two Well, we aren't sure if it's luck or
Yeah.
or care. But anyway, he was very fortunate that he has only 2 phenotypes. And that's not the way the real world works. The real world is way more complicated than that. And what this shows you is, even in Mendel's case, when we add 2 different loci, we go not from 3 genotypes and 2 phenotypes, we go to 16 genotypes and 4 phenotypes. And you can see what happens. That's 1 locus, this is 2 loci, and then this is 3 loci, and eventually what you're seeing is it's starting to meld into a combination, into some sort of mix of phenotypes. And so what that means is we start running into a problem. Whereas before, with 1 locus, we can assign something to a certain class of phenotypes. But when we have many loci, a certain class of genotypes, I'm sorry. So we can take an individual, look at their phenotype, and say, oh, it must want to be one of these genotypes. Whereas at this point, if we look at color, going from red to white in this case, we can't really tell what the genotype is going to be. And of course it gets worse, because we can, for example, have environmental variation. So there are individuals which have a phenotype which lies directly in the middle. And we can't know which genotype to assign them to. And so it starts getting very difficult. Then of course you have complications of multiple alleles. In this case I put up the ABO blood system in humans, where we have 1 locus that has 3 different alleles and can give, as a consequence, even within there, you get a large number of different genotypes. And then, of course, there's our favorite in this class, which is epistasis. These are black Labradors. This is Jim Cheverut's term, which came out of long discussions of various sorts, but he's the first to publish it. He defines epistasis to be the phenotype at 1 locus, well, the physiological epistasis to be when the phenotype at 1 locus varies depending on the genotype of the 2nd locus. So this is the black locus in Labradors, and so if they're big B, big B, big B, little b, they'll be a black lab. If they're little b, they will be a chocolate lab. Now it turns out there's a 2nd locus that interacts with it, which is this E locus. And if you are a big E, or big E, little e, that same rule applies. But if you're little e, you're a yellow lab. And it doesn't matter what your genotype is. And so that's fairly classic epistasis. That's, as I say, physiological epistasis, but there's one of these confusing things. It doesn't seem to have much effect on evolution. And the distinction is that we will be talking about what we could call statistical epistasis. And, I didn't put up, epistasis is interactions among loci that result in phenotypes that can't be predicted from the effects of each gene considered separately. So if we consider these genes, the B locus separately, we say black or chocolate, versus when we add the 2nd one, suddenly we get yellow. And we get individuals like this individual here. If we make 2 of these together, we can get all the possible phenotypes. Okay, and then what you end up with is polygenic inheritance. And this is the classic picture that you get that I like to put up. This is from the Connecticut Agricultural College undergraduates. And believe it or not, they managed to get the entire class, which is very small, and got them to line up in a field in categories. And this is the distribution of those individuals. There's one guy who was 4'8", and another guy that was about 6'5". This was a long time ago. And what happens is, basically you start seeing this normal distribution. This is exactly the distribution you expect when you have a large number of loci, multiple alleles per locus, and gene interaction and environmental effects all coming together in a mix. And we see it not just here. This is birth weight in humans for Norwegians. This is Bill depth, I just sort of randomly chose this. You can see it for all kinds of things, ranging from behaviors to coat color. And so this puts us in an interesting situation. Basically, when we have one or a few loci, we can use Mendelian genetics. And the analogy I put here is an anchor. If we have a boat and we want to anchor it, we can put an anchor into the ground, and that gives us a location on the physical Earth. We know where that is. And you can actually, you can set your GPS so that if it moves more than a certain distance, it will set off an alarm that tells you that you're dragging anchor. But of course, that only, in this nautical analogy, that only works in shallow depths. And we're anchoring on the bottom, and we're positioning ourselves relative to the Earth. Well, the shallow depth, the analogy of Mendelian genetics, is that we're dealing with one or a very few loci. And when we do that, we can anchor to the Mendelian genetics. We can say, what are the individual alleles? What is the locus? What are the alleles? We can identify the effects of specific loci and alleles on a convenient scale. We can just take whatever arbitrary scale we want. On the other hand, we can take the analogy, essentially as you add loci, you can think of that as adding depth to the water that we're anchoring in. And at some point, it gets too deep, and you just simply can't anchor anymore. So what does a boat do when it's in a storm? And there is that problem. They set out what's called a sea anchor. And what a sea anchor is it's a giant parachute which you drag from the front of the boat. And what happens is it drags against the water. And basically, this is what you do in deep water, the analogy being many loci. But what's going to happen is our sea anchor is what we use, and that's quantitative genetics, where we're dealing with many, many loci. But the thing is that we're no longer positioning ourselves relative to the Earth. At least before the days of GPS, you had no idea where you were. But what you could do is you could position yourself relative to the wind and waves. And so that became your new positioning system. How was wind and waves working? The problem with that, of course, is that it's very changeable and it moves as we go on. We're going to do the same thing. We're going to identify effects relative to the population mean. And so that's going to be the effect that we'll be looking for. And anyway, it gets very slippery at that point. And what I mean by slippery is that here we can say that is the gene that does what it does. Here, everything's going to change as gene frequencies change, as the population conditions change, as interactions change. Anything that changes is going to change the population mean. And so it's going to change our results. Okay, so let's take, since I was guessing that mice would be the theme here, the pygmy locus in mice, and we can think about that. It turns out that there are, in fact, two different, pygmy locus has two alleles. The A2, A21, if we take those individuals, and we take a whole bunch of them, any one can have a slightly different phenotype. It will have a different weight, but if we take the average of all of them, they have a genotype, they have a value of 6. So they have 6 grams, they're basically 6 grams. If we take the heterozygote, and we take, again, a bunch of those and measure all of those heterozygotes, we get 12 grams. And if we take the A1 homozygotes and take a bunch of those, we get about 14 grams. And so this is one locus. We can use Mendelian genetics. We can do controlled crosses. And we can get all of these things. Now notice when I say 6 grams, any one individual might be 6.5 grams, 5.8 grams, whatever. But if we take the long-term average, take an average of a whole bunch of them, it'll be 6, 12, and 14. And these are sometimes called genotypic values. And it is a big difference. Okay, so what do we do with this? We have to set up the problem, first of all. And the thing is that, and this is, I'm following Falconer, there's other ways we could do this. What we're going to do is, we're going to take those three genotypes and we're going to take the mean of the homozygotes. Again, we're still in the Mendelian world. And so it's just convenient to take 6 plus 14 over 2, which gives us a fixed point of 10, which is just the average of the two homozygotes without any sort of weighting in there at all. And then from there, it's convenient to put this on a simple to understand scale by just taking the genotypic value, subtracting off the mean of the homozygotes, and that gives us 14 minus 10 is 4. The other homozygote is 6 minus 10 is minus 4. And the heterozygote is 12 minus 10 equals 2. Notice that these are, because we took the average of the two, these are equal and opposite of each other, which will become convenient later on. But notice also that this is not in the middle. And so as a consequence, we can take the population, here's the average of the two homozygotes, and there's minus 4, plus 4, and 2. So that's kind of what we're looking at right here. Okay? And this becomes our 0. But you know, that's kind of something you have to keep in your head, is that we need to add that 10 on if we want to actually get the phenotype. So 10 plus 4 gives us the 14. 10 plus 2 gives us 12. 10 minus 4 gives us 6. Okay. So, there we have it. Now what I'd like to do is, we can actually calculate the population mean. And this is where suddenly things start changing. Because the population mean is not the average, it's the weighted average. And so we're going to take the phenotype times the frequency and add those up to get the population mean. And if we do that, it's A times Q squared, A2 times Q squared, 2PQ times dominance and times the dominance deviation, and P squared times A1. So that gives us a population mean, and we can actually write it this way, because A1 and A2 are equal and opposite of each other, but opposite to each other. Okay, so this becomes our C anchor. This is the population mean that we're going to anchor this to. And you know, we can calculate different frequencies. Here's a frequency of 0.7, and you calculate it through. And this is a frequency of P equals 0.3, and notice that the population mean changes. And it changes dramatically. Here it is 10 plus 1.76 gives us the population mean is 11.76. And here it's 10 minus 0.76, which is 9.324, 9.24, something like that. Okay, so now we need to come up with one of our core concepts of quantitative genetics. And that is the idea of average effect. The average, it's called the average effect of a gene substitution. And this was really the brilliant insight of Sir Ronald Fisher. And he actually defined two terms, the average excess and the average effect. The average effect is, imagine that what would happen is that we have an individual, and we're able to do genetic surgery on it. I don't know what that is, but we can call it that. So we're able to reach in and grab an allele, remove that allele and replace it with the one we're interested in. Okay? So we can take out we can reach in and we can grab one and put back the one we want. So we're interested in the A1 allele. We'll reach in, pull out an allele, and put in an A1 allele. Here's the trick. Our genetic surgery isn't that good. And as a consequence, we don't know which allele we're going to remove and replace with the A1. And so we're just going to randomly take one of the two alleles that's in an individual and replace it with an A1. And if we do that, what we find is, for example, if we take an A1 individual, they have a phenotype of A1. And if we go in and we are interested in the average effect of the A1 allele, we're going to reach in, we're going to pull out that A1 allele, and we're going to put in an A1 allele to replace it. What's the genetic effect of that going to be? Well, it's going to be 0. Okay? And so that's at it. Now, on the other hand, if it's a homozygous A2 allele, and we reach in and we pull out one of those alleles, we will pull out an A2, and we will put in an A1. And so that goes from being an A2 individual with a phenotype of -4 to a A1-A2 individual, a heterozygote, with a phenotype of 2. And so we go from -4 to 2. And so what we see is that that's a change of, it goes up 6. So if we do that calculation, we discover that, this is taken straight out of Falconer, that the mean value of, I was going to say, then the heterozygote. If we imagine we take a heterozygous individual that is D, they have a value of D, and we're going to reach in and take out an allele. Here's the problem. If we reach out in, we could reach in and randomly grab an A1 allele and replace it with an A1 allele, in which case it goes from D to D with no change. On the other hand, we could reach in, grab out an A2 allele and replace it with an A1, in which case it goes from D to A1 to plus A. And so it turns out that the average is just simply the, whether you get, is just simply half of that. So it's, the difference between is D minus A plus 0 over 2. Okay? Anyway, we can weed that down, do the algebra a little bit more easily if we just look at the effects of a single allele and the probability that the individual will be grabbed is A1A1 versus A1A2. And if we do that, we discover that the mean value of the individuals which have had that genetic surgery is QA1 plus QD. PA1 plus QD. Now, we actually can do that genetic surgery if we're dealing with a single locus, because that's what Mendelian mating is all about. That's what sex is. So this is actually what, if we have a single locus world, this is what an individual, what the offspring of that individual would look like. And similarly for the A2 allele, we have, with probability P, they're going to, it will be heterozygote. With probability Q, we get a homozygote. And what happens then is, again, we get this number right here. This number right here. In both of these cases, we have not corrected for the population mean. And so what we do then is we subtract off the population mean, and we eventually end up with these as average effects. And so, math aside, what that's telling us is that what we are actually getting is not what the allele does, but what the allele does in the context of its population, of the population it's found in. Our genetic surgery is Mendelian genetics, and we're doing a cross, essentially, of an A1 individual to a randomly chosen other individual, and you can get these two genotypes out of it. And so we're figuring, so that's how we're doing it. We're saying, what do the offspring look like? Now the important thing is, we can calculate it, and for P and Q, and here's our equation, and this is with exact number, I put, rather than being P and Q, I said, here it's 4 and 2, this is A1, and 2 is D. Here it's minus A1, and, this is A2 plus D, P minus Q. But we can actually calculate it for different frequencies. And we see that the average effect of the A1 allele is 0.96, and the A2 allele is minus 2.24 when P equals 0.7. But it goes from 0.96 to 2 to 3.36. These numbers become very variable as the population as the phenotype of the population changes. As the mean gene frequency of the population changes. And that's the important thing you need to realize. That people who are familiar with molecular genetics always want to go back and say, well, what is the allele? What does the allele do? And now we can't do that. We have to say, what does the allele do in the context of its population? Okay. So this is what a standard quantitative, what we might do with this graph at this point. So here's A2 genotype has a value of -4. Here's the mean of the 2 homozygotes, which has a value of 10. Here's the A1, A2 individual, which has a value of D, or 2. And here's the A1, A1 individual, which has a value of +4, or A1. And so we can actually plot these things. An individual who has A2, A2 is going to be A2, A2 everywhere. The analogy I want you to think about here is that when you look at me, I'm short. I'm 5 feet 5 inches tall. And you know what? I'm 5 feet 5 inches tall, whether you measure me in Peru or if you measure me in Holland. However, if I decide to watch a parade, I get a really good view in Peru because I'm tall. And as an adult, I have to stand in the back, and I can look over everybody's heads and see the parade. But if I go to Holland, where everybody's really tall, then when I try to look at that parade, I don't get to see much because I'm short. And so I'm still the same height. And so what I want you to think about is being the same height is kind of the Mendelian equivalent of, is equivalent to Mendelian. It's measured relative to a fixed standard scale, which is a tape measure. But whether I'm tall or short is a statement about how I sit in the population. Am I taller than the average? Am I shorter than the average? We're doing exactly the same thing here. So these genotypic values are measured on an arbitrary scale. But that's not going to be very useful. You know, when I say how tall I am, it doesn't tell you whether I can see the parade or not. It's, you know, whether I should bring a stool if I want to watch the parade. Well, I have to know not only how tall I am, but how tall everybody else is. So what we want to do then is do a regression of a population through, what we want to do is do a regression of phenotype on genotype, but it's a funny regression. And here I've done it at a gene frequency of 0.5 because it's a weighted regression. And it's a weighted regression which is weighted by the frequency of the 2 genotypes. So here it's 1 to 2 to 1. The population always goes through the bivariate mean. So it goes through 1 and 0, 10. It goes through 1 and 10. And it goes through this point and goes through the, in this case, at a gene frequency of 0.5, it goes through the genotypic values. And so that makes sense. But what happens if we change the gene frequencies? And so here I have 0.7 and 0.3. And so again, the genotypic values don't change. I'm 5'5", regardless of how tall everybody else is. But this individual is mating randomly in the population, and the regression line turns out to be what their offspring look like. So this is regression of phenotype on genotype. And we get a line which goes through the bivariate mean of 12.44. And this bivariate mean, what I mean here is this is the mean gene frequency, 0.7 at 12.44. If anybody challenges me on that particular graph, I did it by eye. so don't. And this is the line, this is a regression line, so it always goes to the bivariate mean, and it always goes, and notice that it doesn't go through the 3 points. And so it's changing with a different gene frequency. Interestingly, it turns out that the difference between these points is A1 minus A2, which is alpha 1 minus alpha 2, which is basically alpha. And the difference between these is exactly the same. So it becomes a linear system. Every time you add an A1 allele, you move up along that line. Of course, it's not really a linear system, right? And that's what made this work. Because I had a dominance deviation in there. The dominance was not exactly in the middle. So if the dominance had been exactly in the middle, this line would have been totally unaffected by gene frequency. It would always go through those 3 points because 3 points determine the line. I mean, 2 points determine a line, and as long as that third one's in the middle, it will just move along that, the mean will move along that line with no effect. But in this case, the dominance is off, this is not on the same line, and so it will move. This is for 0.3 and 0.7. And notice again, the bivariate mean, the number of A1 alleles, is now 0.3, and the mean is 9.24. Again, that is the 10 minus a little bit gets us 9.24. Okay, and what I didn't point out in the last slide is that, again, here we have our 3 lines, and essentially, this is what the offspring look like, and we can ask, what is that value? It actually turns out that the value of this is 2 alpha 2. Okay, so we have the average effects of the alleles. And the breeding value, the mean value of the offspring of that individual is alpha 2. It's 2 alpha 2. So basically, that's what its offspring are going to look like. And again, here's the dominance deviation. It's alpha 1 plus alpha 2. I mean, the heterozygote. And here's the homozygote, which is 2 alpha 1. And so we can actually put those in. Okay. Now, the other thing you might ask is, what about this number right here? And this is the dominance deviation. So it turns out that the genotypic value is the 2 average effects plus the dominance deviation. And the individual of themselves would be a further deviation, which was the environmental variation, which I've left off of this graph. So if we put those two together, this is what it looks like. What we see is that when we go from a gene frequency of 0.7 to 0.3, we get this shift in what the average effects do. And what happens is that the population mean changes, the slopes change, the dominance deviations change, everything changes. And what that change reflects is that these are measures of what the offspring look like. So, why would we do that? The reason we do that is that as we have lots and lots of loci, we can't go in and gel them up and figure out their genotype and make a relationship between the genotype and the phenotype. Now, if you look at the humans are thought to have 25,000 genes. We have literally millions of SNPs, and the majority of those, and we have SNPs which are in coding regions which affect phenotype. But we also, most of the SNPs we find, single nucleotide polymorphisms that we find, are outside of the coding regions, and yet they have huge effects on phenotype. And so we have a really hard problem saying this SNP is going to make that phenotype. It just doesn't work. And so the question is, what can we do about that? Well, it turns out we have another solution, and that's breeding value. The breeding value of an individual is essentially what your offspring looks like. It's actually twice, formal definition is, actually should say twice the mean value of an individual. There should be twice the mean value of an individual's offspring measured as a deviation from the population mean. What it does is it gives us a means of measuring genetics of a population without knowing anything about the genes. This was brilliant on Fisher's part, because back in the 1930s, we didn't even know what a gene was. We just knew that there was that Mendel had identified these factors which segregated, that acted as Mendelian traits, as we call them now. And we knew that there were other ones which didn't. And he provided a mechanism for us to work with that and say, well, what we're going to do is we're going to look at what the offspring look like. That will be our way of figuring out the genetics. Turns out that the breeding value is just simply the sum of the average effects that an individual has. Simply, that gets very complicated very quickly, because average effects are a function of all of those issues we've talked about before. That is, the population mean. We'll see that it's a function of the frequency of alleles at different loci if they epistatically interact, and it can be a function of the environment with such things as diet. All of these things can affect what the average effects will be. So this is the idea. So this gives us our graph, and what I want to get across here is that in fact what we're doing is we're taking the value of an individual. This is supposed to be a normal distribution. I took that dot and smeared it out because this is going to, every individual is going to have an environmental effect on it for the trait we're interested in. If we look at humans, we discover that Japan, I believe, has the largest increase in mean phenotype of any modern population since World War II as a function. You know, if we measure them from World War II to the present. Well, what happened was not a change in gene frequency, maybe a little bit, but that's really not what happened. What happened is that they shifted diets. And they went from a largely agrarian rice-based diet to their modern Western diet, which has an awful lot more protein. And as a consequence, you know, back in 1945, the Japanese were very short people. And today, they're quite tall, and we find that there are, you know, they're very active baseball players. You have to be at least 5'7" to be a baseball player. Do you know why that is? This is kind of fun. The reason that is that the White Sox had a dwarf pinch hitter. And he couldn't hit for anything. But he had a strike zone that was about that big. About a foot off the ground. And so he was good for a walk. And so they outlawed that. So you have to be at least 5'7". I can't play baseball. But anyway, so any individual and any given genotype will be a function, will have some sort of a variation around that, around what their genotypic value is, depending on all kinds of details. Actually, the other story I can tell you about height, which is kind of interesting, is that archaeologists use skeletal height length, how tall ancient people were, as a measure of how good their economy was. Because it turns out that diet affects height. And so if you take the average stature of a population, it turns out that well-fed populations all have roughly the same height. Now you do have things like pygmies who are very short and the Bantu who are very tall, but within Western society, within lots of, you know, as long as you have an idea of what that is, you can really say, you can look at that trend and say, is their economy improving? Was it decaying? And they know that, for example, the stature of individuals of the Aztecs went down prior to the collapse of the Aztec Empire, and it looked like that was a decaying civilization. So anyway, that's a sort of environmental effect that we're talking about. We're going to take an individual's value, we're going to divide it into a bunch of different components. We're going to divide it into genotypic value, its environmental effect, bringing it up to genotypic value. We're going to bring that genotypic value, we're going to divide that into the dominance deviation and the breeding value. Okay, we can do that for any individual in this one locus system, and we could, if we had independent linkage equilibrium populations, we could just simply put as many loci as we want. For every individual, we could put some sort of scale, and we could put their genotype here and do the same sort of division. Okay, so basically what we're going to do is we're going to take the phenotype of an individual, how tall you are, you know, how fast you can run, you know, what flavor ice cream you like. And we're going to divide that into a breeding value, which is what you can pass on to your offspring. You know, it's no secret that I'm short because my parents were short. A dominance deviation, which is the deviation associated with your particular genotype, which is a function of the particular mix of alleles you have, and an environmental effect. And, oops, that E should not. So what we're going to do is, for example, if we look at the A1 individual, any given A1 individual, we're going to divide that into the two breeding values, I probably should not have put, this should be a capital A1. I'm sorry. I wrote this lecture fairly quickly. This should be a capital A1. Is 2 alpha 1, that's the breeding value, plus the dominance deviation associated with the 1-1 genotype, and the environmental deviation that's associated with that particular individual. And so again, this just shows you the same effect right there. Now here's the problem, because I told you what evolution was, right? Evolution is change in a population. And so this is about an individual. And so we need to make that transition to a population. And it turns out that there's a couple of incredibly convenient things. And again, Sir Ronald Fisher was brilliant. One should not lose sight of how brilliant that man really was. And what he did was, remember how he defined everything as deviations from the population mean. So the mean phenotype is equal to the mean breeding value, is equal to the mean dominance deviation, is equal to the mean environmental effect, which is 0. That's really convenient, because what we can do is, we can simply, remember your formula for variance? X squared minus x bar squared? Well, the x bar squared is 0. And so that makes life way easy. Plus, he defined things using regression methods. And so these 4 factors, phenotype, the breeding value, dominance, and environmental effect, are what we call orthogonal. They're independent of each other. And so that's beautiful, because it turns out that the phenotypic variance is due to the additive variance, plus the dominance variance, plus the environmental variance, plus a whole slew of covariances that equal 0. So this is one of those cases where there's this gigantic part of this equation out here that equals 0. And it's beautiful. Okay. Also, remember that I'm ignoring higher order interactions. That will be something that we will definitely be relaxing as this course goes on. Okay, now let's do that right now. This is just, I'll put up all of the different kinds of epistasis later on. But this just gives us my formulation of 2 different kinds of epistasis. Jim Cheverud will give us, uses somewhat different partitioning. They're both valid. I like mine. And I categorically reject his. But this is, let's see, should I? Yeah, okay. We'll just make a brief forward and then go back into the why we care about this. This is additive by additive epistasis. Now, why do I call it additive by additive? The A locus is additive, but the direction depends on the what genotype you have at the B locus. And the B locus is additive, but the direction depends on the genotype at the A locus. What I mean by additive is, if we look at those, here's A1, A2, A2, okay, I got it backwards. Here's A1, A2, A2. And notice that the dominance deviation is 0. That's what I mean by additive. Okay? The dominance deviation is exactly intermediate. This is additive by additive, and just for fun, I put a additive by dominance, just to show you what's going on here. Sorry, dominance by additive. The A locus here is over-dominant, neutral, or under-dominant, depending on the genotype at the B locus. And the B locus is additive, but the direction of additivity depends on the genotype at the A locus. So that's what we mean by, that's sort of the logic behind naming them. But anyway, let's look at one of these. How would we do that same regression in a 2-locus system that I just showed you? And we can actually do that. Oh, here it is. The A locus is additive, the B locus is additive, and there's additive by additive epistasis. And we can actually do the same regression, except it turns out that it now becomes a three-dimensional plot. And so here we have the A1, A1 homozygote, heterozygote, and the other homozygote. And here's a B1, 1, B1, 2, B2, 2. And so we see that the A1, A1, B1, B1 has a value of +1. The heterozygote has 0. And the other homozygote has -1. So it's additive within that genotype. Here it's neutral within the B1, B1, B2 locus, and it's additive in the presence of the B2, B2 locus, but the opposite direction. And we can do the same thing. Here's the B1 locus, additive, neutral, and additive going the other direction. Now when we do the regression, if we do the regression at a gene frequency of 0.5, it turns out that there's no additive genetic variance, that this population is sitting at a point where it does every one of those alleles is neutral. That regression gives us a flat plane. And so if we wanted to do 3 loci, we'd have to have a 4-dimensional graph with fitness on one axis, and the other 3 axes would be the A, B, and C locus. So my brain isn't that big, and so I limit myself to 2 loci. But anyway, here's the interesting thing is, let's change the gene frequency of the A locus from 0.5 to 0.25. So we're going to put it right here. So here's the gene frequency, and at the B locus we're going to leave it at 0.5. And so now our trivariate mean is here, here, and somewhere over here. Look what that does. Now when we do the regression, this B genotype, in the presence of the A2 locus, gets a lot of weight, and the A1 locus gets very little weight. And what that does is it tips the B locus. So when we change the gene frequency of the A locus, we change the average effects of the B locus. And that's going to be the important thing that's going to come out as we move along, is that in fact, it's not just your gene frequency, it's your interacting partner's gene frequency that affects your average effects. So anyway, that's why I say we can move these complications up. I left the complications out, but we can put them in. Okay, so if we want to add all those things in, we're actually going to have to divide the phenotype into the breeding value plus the dominance deviation plus the additive by additive epistasis plus the additive by dominance epistasis plus the dominance by additive epistasis plus the dominance by dominance plus the environmental interaction. And you could keep adding, additive by additive by additive and additive by dominance, and as far out as you want to go. There's statistical reasons why there's not much point in going beyond 2 in many cases. But again, they're all defined orthogonally, independently of each other, so we can transfer that down. What we're doing is we're taking the phenotypic variation in the population, something that is quite measurable. We can line everybody up in this room and measure how tall every individual is. And that would be our phenotypic variance. And then if we did some sick breeding design, we could divide it into additive effects, dominance effects, additive by dominance by additive, etc. And we would divide it into all of these different variance effects. Now, remember that what we're doing is that these are a function of the population and the individual's genetics. So what that statement would give us is the partitioning of that phenotypic variance in this population. And if we looked at a different population, if we did the same thing in Finland, we would get a different answer. So, why do we do this? Why would you, this just seems so stupid to me at this point, right? Because what we're doing is we took this perfectly good system where we said, we know what the allele is, we know what the allele does, and we could just simply go out and say, oh, that's the gene for breast cancer. and be done, or what have you. And instead we've got this thing which is completely variable. Depends on the gene frequency, depends on the population mean, depends on gene frequencies at other loci, depends on environmental effects. Why do we care? Well, we care because of VA, because of the additive genetic variants. And it turns out that the additive effects are the genetical effects that can be passed from parent to offspring. That's by definition, right? That's what we did. The breeding value is what your offspring look like. And so by definition, that is what the additive effects are. The additive genetic variance is the variance in those effects that can be passed from parent to offspring. And this is the important trick, is the additive genetic variance is a variance that can contribute to evolution by natural selection. And that's why we care, because what quantitative genetics does is it allows us to predict how a population will respond to selection. Okay? Now in this class we're not going to get into how you actually measure additive genetic variance. It turns out that Fisher's original formulation is, his original definition, believe it or not, is the covariance between average effects and average excess. And nobody knows how to measure average excess. Remember, average effects is the genetic surgery. Average excess is what the mean phenotype of an individual who contains that allele is. And it turns out there's a subtle difference between the two. And nobody really knows how to measure those. So there is a real sort of fundamental problem. But nevertheless, we can measure additive genetic variance in any population by doing appropriate breeding designs. And one breeding design we could do, sorry, I don't have it there right now. One breeding design we could do is to simply do a regression of offspring on their parents, or more commonly, offspring on their mid-parents, the average of the two. The other thing we can do, again, beyond the scope of this course, is a half-SIB breeding design. It turns out that ANOVA uses an F statistic because the guy who invented it, his name started with an F, Sir Ronald Fisher. And he invented it specifically as a way to get at genetics. And so it turns out that your standard nested design can be used to figure out what the additive genetic variance is. And so that's how it's measured in a practical manner. I'm not going to tell you how to do that. Okay, so anyway, it turns out that the additive genetic variance is so important that we actually have a term that we use with that, which is the heritability, which is the ratio of the additive genetic variance and the phenotypic variance. And it turns out that the additive genetic, that ratio is, as I say, fundamentally important. There's actually another interesting piece of history. H squared is not H squared. It's H squared. And for those of you who don't speak English that well, I'm making a distinction that is a single symbol.